Hello everyone, my name is Lily and I'm a CSURP student from the Structural Biology Initiative. This summer I had the opportunity to work in the Daniel Kitty lab with my mentor Blake on an exploratory project that studied allostery across the protein tyrosine phosphatase family. Before I begin talking about my research, I will briefly introduce what protein tyrosine phosphatases are and why we want to study their allostery. Protein tyrosine phosphatases, or PTPs, are a family of enzymes that regulate processes like self-death and self-replication. In fact, our human genome encodes over 100 PTPs, and the dysregulation of PTPs has been shown to be linked to various diseases such as diabetes, cancer, and Alzheimer's. However, PTPs are regarded as undruggable because PTPs are very similar around the active site, as you can see in Figure 2, which make it difficult to target one specific PTP and not affect another. Fortunately, PTPs are very diverse at other regions outside the active site, the allosteric sites, so it may be easier to target allosteric sites for the development of drugs. Interestingly, the allosteric sites that have already been discovered were found to be clustered together as shown in Figure 1, which suggests a common allosteric framework. While allostery is a widely accepted phenomenon, little is known about allosteric mechanisms and the transmission of allosteric information, which led us to conduct an exploratory study that studied PTP allostery. In order to search for hidden allosteric networks, we followed a series of steps outlined in this slide. We first began by scouring the Protein Data Bank, which is a worldwide repository of original X-ray diffraction data in the respective crystal structure models. From this archive, we were able to gather information about 88 PTP crystal structures from 25 different types of PTPs. Once we had a wide variety of PTP structural models, we moved on to step 2, in which we selected 6 structures that would be compatible for comparison. In step 3, we searched for unexplained electron densities in the electron density maps and manually modeled missing alternate conformation using a modeling program called CUT and a refinement program called Phoenix Refine. In step 4, we auto-modeled additional conformation using a program called QFit. And in the final step, we look for potential allosteric networks, and particularly those that connect allosteric sites to the active sites. Now that I have shown you the big picture of my research project, I will walk you through the details of each step and also share some preliminary results we obtained throughout this entire process. Figure 3 nicely summarizes what kind of work we did in step 1, which was gathering data about various PTPs from the PDB website. This histogram shows a distribution of the crystallographic resolution of 88 PTP structures. In crystallography, lower angstroms mean higher quality X-ray crystal data, so we aim to select structures with lower angstrom values to move into step 2. These are the structure models we ended up selecting in step 2. When comparing X-ray crystal structures, it is important to minimize the number of differences in the experimental setup. Therefore, we carefully selected 2 sets with closely similar conditions. The members of the first set are all one type of PTP, but vary in space groups, and the members of the second set have the same space group but vary in PTP type. In step 3, we looked at unexplained electron densities and remodeled alternate conformations in a program called CUT. Our goal was to improve the currently available model by adding missing conformations and minimize the green and red blobs shown here on the left, which respectively represent missing or excess electron densities. In this step PTP structure, we modeled an alternate conformation of the cysteine residue as shown on the right. As we updated the electron density maps with Phoenix Refine, we can see that the green and red blobs have minimized. This model is a better fit to the experimental data. Similarly, in this PTP epsilon structure, we remodeled residues that were previously missing in the structural model. Here again, we can see that the green blobs have minimized after updating the electron density map with Phoenix Refine. Once we completed manually adding alternate conformations to the PTP structure models, we used a program called QFit to automatically model residues. QFit is a program my mentor is developing that is able to find many more additional residue conformations that have weak signals in the experimental data. In Figure 7, the image on the left shows how the structural model of PTP N18 looked after we manually modeled alternate conformations, and the image on the right shows how the structural model looked after we auto-modeled alternate conformations with QFIT. Although we haven't fully analyzed this data yet to identify allosteric networks, this preliminary data do seem promising in the sense that we can already see some potential residue networks that connect the allosteric sites to the active sites. In step 5, which is the final step, we examined our updated PTP structures to search for residues that suggested the presence of allosteric networks between sites shown here in Figure 8, which shows the allosteric sites from various members of the PTP family. Although we haven't analyzed our data thoroughly enough to conclude the presence of allosteric networks in the 6 PTP structures we studied, we believe that continuing with this process with many more PTP structures will lead to the discovery of new therapeutically relevant allosteric networks and ultimately improve our understanding of allostery across the PTP family. The data I have generated and shown you today highlights the presence of many unmodeled alternate conformations in crystallographic data. For future directions, this data will be analyzed by the CUNY lab to search for networks of alternate conformations that bridge the active site with known allosteric sites. This will shed light on mechanisms of allosteric transmission. Thank you everyone for your attention. I would also like to thank CSURP and the Structural Biology Initiative for hosting me this summer. A special thank you to my mentor Blake, who has gone extra lengths to teach me, and also a special thank you to my PI Dr. Daniel Keaty for welcoming me to his lab once again for another great research experience.